Okay, so you can just share your slides then, and I will start. Okay, fine. Very good afternoon to everyone, and welcome to Vertical 2 session of the DDH 2020 training program. Today for this session, we have with us Dr. Gopal Lal Khatik. Dr. Khatik is currently working as an assistant professor in Department of Medicinal Chemistry at NIPER Raebareli. Dr. Khatik is a pharmacy professional and working on computer-aided drug design via exploring small heterocyclic scaffolds. After graduating in pharmacy, he earned MS and PhD in medicinal chemistry from NIPER Mohali as a NIPER Fellow and Fellow of Engineering and Technology UGC. He worked in the industry and academia for 8.5 years at various positions. He has been published 54 research papers and review articles in index journals of high repute like ASC, RCS, Wiley, Springer, etc. He has also edited the guest issue of Current Drug Discovery Technologies and Current Pharmaceutical Designs for Pantham Science. Also, he has been authored a book on medicinal chemistry for BPharm. He has been awarded as SERB Young Scientist and other research awards from various organizations. He is a lifetime member of various professional bodies. Dr. Khatik has expertise in computer-aided drug design and small molecule synthesis. Its research interest includes heterocyclic chemistry, asymmetric synthesis, and drug design in the area of cancer, diabetes, and neurodegenerative diseases. In today's session, he'll be talking about fragment-based drug design. So I now hand over the session to Dr. Gopal Lal Khatik.
Okay, thank you, Dr. Hridoy, for your kind words. So I welcome all the audience who are actually connected online for this particular session, Vertical 2. And I'll be discussing today about the fragment-based drug design, a tool for design and discovery of lead compounds. So before I'll discuss about the fragment-based drug design, I would like to introduce about drug design, what actually drug design, and that will help the beginner to understand how drug design is very essential or important in today's scenario. So drug design is a process to generate and modify the molecule by prediction whether a given molecule will be binding to the target or not, and if it is binding to the target, how much that is powerful. So generally we used to design the drug based on, like, say, traditional method as well as computational drug design methods. So drug design, basically, if we try to understand the process, it involves a lot of efforts, money, and as well as time. So millions of US dollars are being invested to develop a single drug, and that is very costly, of course. And that involves the different processes, beginning from the high throughput screening, thereafter pharmacological screening of the thousands of compounds, and that goes under preclinical and then clinical evaluation before coming into the market. So if we try to calculate the time and efforts required to design a single drug molecule, it takes generally an average of 12 years to accomplish the approval of the drug. So where this computer-aided drug design can help us to minimize the duration of time as well as the cost involved. So how this computer-aided drug design is helpful in, like, say, facilitating the drug design process. Generally, if we consider and simply compare between the traditional drug design as well as the computer-aided drug design, in case of traditional drug design, is somehow random and based on the trial and error. So we don't have much logic behind the method which adopted in the traditional drug design. Also, that is very time consuming and very expensive as well, because a lot of efforts are being put in the screening, synthesis, modification, and thereafter preclinical and clinical studies. And that's how it becomes low yielding. Somehow, 1 in a million molecule can reach to the uh, drug or in the market as a drug or in the market. So in this case, computer-aided drug design can help, as I told you earlier, uh, because there is some logic behind the computer-aided drug design. It's very much specific for a particular target or a particular ligand or a molecule. So it becomes easier us to design the relevant molecule. So it means that becomes faster. And nowadays we have automatic computer-aided drug design software, which also lead to decrease the time as well as the cost. And success rate is comparatively higher in comparison to the traditional drug design. So traditional drug design is replaced nowadays with a virtual drug design approaches, or we can call them as a computer-aided drug design. Or sometime we also call them as a in silico drug design approaches. So it's a very powerful or emerging alternative replacing the traditional based drug design approaches to discover the new lead compound and later on to make a drug. So it's acting like a free filter for the drug design process, because we cannot replace the other processes like say preclinical or clinical studies. But at the initial stages where we used to do the high throughput screening with the help of the pharmacological screening, there we can minimize the drug molecule using the pre-filtered molecule. So this virtual drug design is generally based on the either ligand or either protein. So we can classify them in broad way in two categories. One is a ligand-based drug design, another is a structure-based drug design. Ligand-based drug design is commonly used when we don't have the information or structure of the protein, whereas the structure-based drug design we use when we have the information or structure of the protein or target. So in the case of the ligand-based drug design, different software or programs are being used, QSCR, pharmacophore modeling, and virtual screening. Whereas in structure-based drug design, we use the docking de novo drug design. So our today's talk is very much focused on the fragment-based drug design, and this fragment-based drug design is used in both the ligand as well as structure-based drug design approaches. So these are virtual drug design approaches. Somewhere fragment-based drug design initially used for on the basis of the experimental processes like say NMR, XRD, and ITC, or similar kind of experimental approaches. So now, of course, we are actually looking for the alternate to the experimental drug design approaches to the computer-based drug design approaches. So in computer-based drug design approaches, one may ask how we go forward. Generally, if we don't have any idea what are the general rules for finding out the better drug-like molecule. So in this direction, already we know about the Lipinski's rule of 5, which actually discovered by the Lipinski in 1997. So that says about the pre-filtering of the molecule on the basis of their molecular weight. Okay, so molecular weight must not be more than 500 Dalton, and hydrogen bond donor must be less than 5, hydrogen bond acceptor must be less than 10, and partition coefficient or log P value must be less than 5. So all these are the multiple of 5. So that's why this is known as the Lipinski's rule of 5. These are also known as the Pfizer's rule or R of 5. So these type of rules are generally nowadays considered as the thumb rule for pre-filtering of the drug-like molecules. That gives you idea whether this molecule may act as a drug or not. So Lipinski suggested that may not be all parameters are being fulfilled by the particular molecule, because we can see in literature also so many molecules which are having the molecular weight more than 500 Dalton also are the drug molecule. Like say if we talk about the antibiotic, most of the antibiotic or the certain antibodies or different proteins. So all are having the molecular weight 500. But he emphasized, like say, generally when we are looking for the small molecule, we must consider all these parameters. And these parameters generally he observed based on the pharmacokinetic of a molecule. So if we start with a one particular molecule which is having the higher molecular weight and having the more number of the hydrogen bond donor and acceptor, having the higher log P value, that may lead to the consequences later on. So we observed that most of the molecules later on failed in the clinical studies because of the issues of the pharmacokinetic profile that may having the low absorption or low distribution, low bioavailability or high rate of metabolism or excretion. So therefore, he suggested that if a particular molecule is is obeying the at least two rule, or is not obeying the at least two rule, is not worth to carry forward, because a lot of efforts are being involved in the later on in carrying out the pharmacological screening. So he suggested that at least two rules must be obeyed by the small molecule, to be a drug-like molecule. If all four are being obeyed, it's well and good. So I'll be coming on to today's topic, that is a fragment-based drug design. In literature, you can see a lot of research is being done and published on the basis of the fragment-based drug design and drug discovery. So this fragment-based drug design is also known as a fragnomics. And in one of the article which is recently published in 2018 in APS journal, shows the importance of the fragment-based drug design. So if you see here, this is the comparison between the fragment-based drug design FBDD and other drug design approaches. So overall, you can see here, it's almost 50% or maybe 1-to-1 ratio of the FBDD with the other drug design approaches. So it means this fragment-based drug design approach is very much fruitful, and I'll show you in a later slide how the different drugs are being introduced in the market within a short span of the time period using the fragment-based drug design approaches. So another research which is being published and shows that this kind of approach, FBDD, can be used in the different targets or different classes of the protein, including kinase, proteases, different enzymes, PPI, GPCR receptor, or different kind of transcription factor. So these data are being retroanalyzed from 2015 to 2017. So it means this is tremendously increasing. And there, in the case of the fragment-based drug design, to develop the fragment into the lead molecule, different approaches are being explored. So it is not always like, say, only computer-aided drug design is being used. So as I told you earlier also, the different methods which are based on the experimental methods are also used in the fragment-based drug design, like say NMR or the other biochemical assays, pharmacological screening, or XRD, ITC, or different approaches, miscellaneous approaches can be used to find out the lead molecule from the fragment-based drug design approach. So I'll be coming to explain you what is the actual fragment-based drug design. I showed you the importance how this FBDD is growing in today's scenario. A lot of publications are coming around, and overall you can see 3 drugs are being approved by the FDA, and 30 drug derivative or 30 molecules are under the clinical trial. So fragment-based drug design, you can see in this picture, is a very powerful tool, and it's a rational drug design approaches. So initial fragment library is being used to target the particular protein receptor or drug target. Among these, all are being optimized at the binding site, and later on, all these fragments are being linked together to give the final structure or final molecule as a lead molecule. So as I told you, this approach is very much beneficial in reducing the cost as well as time. And as I told you, currently 3 drugs are being approved by the FDA, and first drug which is approved by the FBDD is vemurafenib in 2011. So I'll discuss one interesting example here. You know about the Gleevec, and this Gleevec is actually a molecule of the Novartis, and in recently in 2013, its patent is being denied by the Supreme Court of India, which is filed by the Novartis. So Gleevec is initially comes as a brand name in the form of mesylate salt of the amitinib. This amitinib is an anti-cancer drug. Okay, and this amitinib is being discovered based on the virtual screening. And this virtual screening is based on the fragment-based drug design. So fragment-based drug design, where library of the fragment is being identified for a particular target, and those fragments are being optimized later on. So you have a lot of molecule as a fragment in a library. Among them, generally a molecule which is having the low affinity or low efficacy will be selected first. Later on, those molecules which are or fragments which are having the low affinity or low efficacy are being optimized at the binding site. So I'll show you this, how the different fragments are being selected. It's similar to the puzzle how we solve simply joining the different composites. So It's based on the same phenomena. So first we have to identify the relatively feasible fragment which is going to bind to the active site. Thereafter, these are being selected and these are being trying to connect together. So connection may be somewhere hit and trial method, and that is being done through the virtual screening. So this connection approach is based on the which is going to fit in a better way. So all possibilities are being tried, and later on a better fit is being observed by the software itself. Or somewhere, if we are using the experimental based drug design, then the characterization is the important part, and that is being done on the basis of the NMR, IR, to find out the dissociation constant of a particular fragment or a molecule on the binding site for a better comparison. So this end up in the Gleevec molecule. So this is how the connect, all those are being, or fragments are being connected to find out the final drug molecule, that is the Gleevec imetinib. So there are a lot of examples, interesting examples I'll be discussing in the next part of the presentation. Before that, I'll be discussing first important part is what actual fragments are being defined. Like say you may have a curiosity, which fragment is important and how we define fragments. Generally, fragments are being defined as a low molecular weight scaffold having the moderate lipophilicity but having the high water solubility. Why it is so important, say water solubility? Because these fragments are having the low efficacy, so we try to test them in the higher concentration. So if they are not having the good solubility in white water, the screening would be difficult. So these fragments, as I told you, must be having the low molecular weight, low lipophilicity, but having the high water solubility. And they must be, as I told you, they must be having the low affinity, which can be in the range of the millimolar or micromolar. And why this is so important? Because later on we will be connecting the different fragments, and thereafter that may show the potent efficacy. Okay, so they are more efficacious later on and can be designed in the better lead molecule, which is having the nanomolar efficacy or affinity. So generally these fragments are being either grown. Okay, so one fragment is initially connected with another, or maybe linked by the different approaches, or maybe merged. So if something is common, we can remove that part and connect them together in the form of merging. So here also, generally we follow the rule of 3. So rule of 3 is actually again a thumb rule, but not essentially followed, but we have to consider them. So rule of 3 says about the molecular weight must be less than or equal to 300 Dalton. So this is less than the 300 Dalton. Hydrogen bond acceptor and donor must be less than 3, and log p value also, or you can say partition coefficient also must be less than 3. Whereas the polar surface area is equal to or less than 60 angstroms square. So these all properties are actually very similar or an extension of the rule of 5 or Pfizer rule. So, for example, if we have the 2 fragment or 3 fragment, while connecting them, they automatically their molecular weight will be increased. Their hydrogen bond acceptor donor will be increased. All of P will be increased. So therefore, these are being limited to the some value, and thereafter, once you connect it, that will be obeying the Lipinski's rule of 5. So this is actually recently evaluated and published in the Journal of Medicinal Chemistry, like say what actual properties uh, carried by the fragments or the lead molecule. So these authors have been collected the 85 examples from the literature, which is published in between 2015 to 17. And they observed that the first plot, which is showing the molecular weight range for a fragment, if you can see here, the molecular weight range is limited to the near around 300. And the maximum number of the fragment are in between the molecular weight of 200 to 250. While in the case of the lead molecule, which is, you can say, almost close to the drug molecule, is having the molecular weight almost like say equal or less than 500. There are some example which has the molecular weight higher than the 500 also. But most of them lead molecule, most of them are having the molecular weight near around 400. Similarly, the partition coefficient calculated log P for the fragments, this plot B shows the comparison for the fragment and the lead molecule. Fragments are generally having the, like say, this is the maxima, and this shows the fragment are having the log P value near around 2. Whereas the drug molecule are having the near around 3. Okay, and if we correlate the log P value with the Lipinski's rule of 5 and rule of 3, these examples prove that most of the fragment and the lead molecule are obeying the Lipinski's rule of 5. Or for the fragment, we actually follow the rule of 3. So now coming to the next part, where they also compared the planarity of the molecule. Fragment, as I showed you initially in the example of the Gleevec, most of them are planar. So fragments are mostly follow the planarity, and they are very much planar, and their RMST is near around 0 or 0.2. And even if we try to compare the other fragments also, though totally there is a RMST near around 0.8. So most of the fragment are almost planar. And the deviation, of course, but that is not much high. In case of the drug molecule, the lead molecule, of course, the planarity is somehow disturbed because of the linkage. So you may not have seen here the much planarity for the lead molecule, which tends to be later on as a drug molecule. So in this case, they also cover the different type of the target protein, and they found that most of the fragments are following the planarity. Some of the are not following the planarity, but of course in the range of the planarity, that is 0.2 RMS to 0.8 RMS. Now coming to the actual process of the fragment-based drug design. So I'll be explaining here how the fragment-based drug design is used to find out the lead molecule out of the fragments. So generally it involves the 5 steps. So it begins with the fragment library. Okay, so this is how fragment libraries is being either uh, designed or either prepared. So designed and prepared or maybe commercially obtained. Now these fragment library must be having the somehow target specificity. So in other approach, like say if we are using the computer-aided drug design, we can simply take the different fragments and try to find out the hit molecule. Now these hit molecule in the case of the computer-aided drug design, hit molecules are generally we consider which are having the either low affinity or you can say particular efficacy but not much potency. So these hit molecules initially are being characterized. In case of computer-based drug design, we use the molecular docking or the other method like say de novo drug design. But in case of the experimental, we use the NMR or ITC or surface plasmon resonance. So all these techniques, along with the X-ray crystallographic study, are being used to characterize and find out the affinity or dissociation constant of a particular hit molecule toward the target molecule. So in other way, it could be through the high throughput screening. So high throughput screening where millions of the compounds are being used. These millions of the compounds or fragments are being converted into the hit molecule. Again, they are being characterized for their affinity or their dissociation constant. And later on, those are being linked together to find out the particular structure. These structures are being optimized in the way to get the better binding affinity or better dissociation constant. Better means lesser, okay? So dissociation constant, which is less, means tightly bound. And in later stage, these fragments to hit are being also optimized based on the SAR by catalog. So in this case, whatever commercially available molecules are there, which are having the similarity and having the somehow efficacy are being used as the fragment to convert them into the hit molecule. And later on, finally, based on the same like the characterization, one can identify the lead molecule which is having the affinity, higher affinity and potency in a nanomolar range. So this is a formal process of the fragment-based drug design. And basically, based on the target molecule or the ligand, we can use the different approaches for the optimization as well as characterization. So basically, experimental approaches which are used to find out the interactions are, one is the NMR technique. So different NMR techniques are being used, like say ligand observed NMR or protein observed NMR. In ligand observed NMR, like say STD, which is this saturation transfer difference, and another is Waterlogsy, ligand observed, gradient spectroscopy, CPMG. So these actually help us to find out the ligand and protein interactions. So particular molecule will be taken, like say, which is having the affinity, non-affinity. The same molecule is being treated with a particular protein, and an unknown molecule which is being also added to the solution in the presence of the saturated amount of the known ligand. So this actually shows the differences in the chemical shift of the particular amino acid residue where the ligand shows the interactions. So other option is the protein observed NMR, where HSQC, heteronuclear single quantum coherence, also known as a single bond correlation, is being used. So we know about, like say in protein, the nitrogen and the hydrogens, which are polar in nature in case of the amide bond. So this can be used as the technique for finding out the interaction of the ligand with these polar bonds, like the nitrogen and hydrogen. So hydrogen, nitrogen, HSQC, or hydrogen carbon, but hydrogen, nitrogen, HSQC is commonly used instead of the hydrogen carbon, because hydrogen, nitrogen bond is somehow more polar and give the better results. So whatever ligand is having interaction with such a hydrogen and nitrogen amino acid residue or bond will be reflected by the HSQC. So a difference between the chemical shift is being observed. So that shows the how strong a particular ligand is interacting with this polar bond. We can use X-ray crystallographic studies also. There are two techniques where one is a co-crystallization, another is soaking. In co-crystallization, protein ligand receptor, protein ligand complex is being formed, and that is subjected for the later on single XRD study. Whereas in soaking, a high concentration of ligand is added in the protein crystal. So the XRD will be showing the what actually interaction where this particular ligand is interacting. But this process is very costlier in comparison to the NMR. We can use the other technique also, like say, as I told you, the surface plasmon resonance, and that actually tells you when ligand bound to the protein surface, the molecular weight is going to change. So that will be detected by the help of this technique, SPR. We can use the different bioassays also, enzyme binding assays also for detecting the interaction. As I told you, we can use the ITC also, isothermal titration, calorimetry. This technique is very useful, particularly for the nucleic acid and as well as other macromolecules like say protein. So this tells you, like say, once the ligand bind with the protein, it changes the thermokinetic properties, and thermogram will tell you whether it is actually having the affinity or not, and how strongly it has affinity. So by comparing the thermogram obtained from the ITC, we can determine the interactions of the ligand with the protein. So this is what I discuss about some experimental method for the characterization while we using the fragment-based drug design. But optimization, as I discussed earlier, optimization is very important part of the fragment-based drug design or fragnomics. Optimization is being done on two ways. One is the growing, another is the linking. So in growing, first we have to identify the one fragment, okay, and the another fragment is being linked directly to the first fragment. So we have to find out, like say in this picture, initial one fragment is selected and its affinity, or you can say dissociation constant, is being determined. Also, you can calculate the LE, that is the ligand efficiency. Ligand efficiency denotes that the free energy observed while binding of the molecule to the target site with a per heteroatoms, like say heteroatoms which are other than hydrogens. So that is later on connected, and again the dissociation constant L is being determined. Then again it is being grown to the other side, and again this dissociation constant is being determined. So you will get here a template. This template later on is being again modified to find out the better molecule or lead molecule. So from fragment to the lead, this is the growing approach where you start with a 1 point, and after starting, you just connect the dot and go to the other point. In other approach, the fragment linking, this is another approach where you will be finding out the fragments at the different site first. So it's a example here, two basic sites is there in the receptor. So two fragments are being initially identified, which are based to fitting with a better KD value. Then these are being linked together with some linker, and later on these linker and other side chain is being optimized. So that lead to the final optimization of the lead molecule. So these two basic techniques are being used in most of the fragment-based drug design approaches, whether it's through the like say computational, or maybe like say through experimental. So I'll discuss first example, a very interesting example, which is based on the fragment-based drug design. So it's begins with the NMR studies. So NMR, is a very important tool for the characterization of the different protein, small molecule, and their interactions. So this is explored in the form of SAR by NMR for targeting the FK binding protein. So initially, it started with a search of the different fragment which are binding to the one side of the FK protein, FK binding protein. So this is trimethoxypicolinonic acid is being initially identified with having the dissociation constant 2 micromolar. And later on, this is the another moiety, which is anilinamide, is being identified with again same micromolar with a micromolar activity. So initial fragments are being identified based on the NMR, and later on those are being optimized by linking a particular linker as shown in the structure beginning from the 10 to 40. So you can see here proton NMR, which is the hydrogen-nitrogen HSQC of the FK binding protein in absence and presence of the compound 3. Okay, so this is a compound 3 initially identified having the R1 hydroxy or benzenilamide, having the R1 hydroxy, R2 hydroxy, R3, R4, and R5 as a R3, R4 as a hydrogen with a 0.8 micromolar activity. So this is in presence of the 3, the interactions is being identified, and this shows that this 3 molecule is having the some interaction because these labeled residues are being observed significant shift in the chemical shift of the proton. So you can see here glycine 51, glutamic acid 54, glutamine 53, so aspartic acid 61. So these amino acid residues are having the interaction with this benzenilamide, and therefore we can see a drastic or significant change in the chemical shift. So later on, these two are being optimized by varying the different substituent, and that comes up with a molecule 9, which is having the R1 hydroxy and R4 as a hydroxy. So para position on both sides. Para position are being occupied with the hydroxy showing the 0.1 in micromolar activity. So these are being linked because we get here idea about one fragment this and another fragment this, and when these are being linked with a possibly what approach we use generally, which is having the chemical reactivity will be linked first. So chemical reactivity here we have a ester and here we have a hydroxy. So we have a hydroxy on both sides. So both are being tried. And among them, this one connection showing the better range of the activity that is in a nanomolar range. So different connections are being tried over here, varying the carbon linker, where number of the carbon in between these two molecules varied from 3, 4, 5, 6, or somewhere else option is also being tried. So it shows that the optimized structure 10 showing the 19 nanomolar Kd value or activity. So this is also actually identified with the help of the NOE experiment, nuclear over-Hauser effect, which is intermolecular interaction in 3D. So this intermolecular interaction shows the better binding of the molecule at the two different locations, like say 2 and 9. Okay, so 2 and 9 initially is being observed through the NOE also. So both are saturated, used in the saturated concentration in presence of the FK binding protein. So this is how NMR lead to the discovery of the new FK binding protein, which is somehow showing the good nanomolar activity and is now under clinical trial as an immunosuppressant. Now, what I discussed earlier is the drug which are initially uh, screened or identified based on the fragment-based drug design approach. But this is the interesting example which I'll be going to discuss is the first approved drug by the FDA using the fragment-based drug design. So this is a Vimura-Feni, and this is sold as in the name of the gel border. And this is very useful and anti-cancer drug targeting to the BRAF. It shows the dramatical efficacy, particularly for deadly form of skin cancer. This drug is developed by the Lexicon in partnership with the Roche. Now this Lexicon is overridden by the Daiichi Sankyo. So I approved this drug in 2011, and this interesting story started with a azaindole moiety. This azaindole moiety was initially having the lower efficacy near around IC50, near around 200 micromolar with a ligand efficiency 0.56 for the different tyrosine kinase protein, like say proto-oncogene protein. And it means that is less selective and having the multiple crystallographic binding mode. So by using the fragment-based drug design, this is connected with the help of the growing technique, connected with the aniline moiety, and this shows the somehow good efficacy in comparison to the azaindole. 100 micromolar activity is being shown by this compound 1. Later on is being optimized by same fragment-based drug design through the growing approach. So different fragments are being added, like say in the azaindole, 1-chloro is being added. Then later on the other fragment is connected with the carbonyl rather than the amine and sulfonamide linker. But this linker is not actually a side chain. So side chain is being added as the sulfonamide, and this lead to the development of the PLX 4720. And this shows the somehow selectivity toward the BRAF protein, mutated protein, but it's somehow non-selective also. Selectivity is there, but it is also acting on the other proto-oncogene protein or other kinases protein. So later on, it is again optimized with the help of the growing approach using the fragment-based drug design, which led to the umorafenib having the high efficacy, although that is lesser than the PLX 4720, but having the much selectivity. Okay, this is much more selective toward the BRAF mutated oncogene protein. So this lead to the successful story of the vimurafenib as an anti-cancer drug, and this drug is being developed only in 6 years. As I told you initially, an average 12 years is needed for the approval of the drug from the beginning. But this is almost half of the average timeline for the approval of the drug. It means we are able to save the at least 6 years to develop the drug using fragment-based drug design. So it is very rapid and evolutionary approaches. Now I'll be discussing one more interesting example, and that is a BCL2 inhibitor used for the chronic lymphocytic leukemia. So here the link approach is being used for the fragment-based drug design. So initially 2 fragments are being identified, fragment 1 and fragment 2. So they have the activity in micromolar, millimolar range, and those later on linked via the linker, that is the double bond between this biphenyl, one biphenyl and another biphenyl. So different linking are being tried, but this was the optimized one, which is having the activity near around 1.4 micromolar, better than the initial fragments. So later on, this is being optimized again by linking approach, which lead to the navitoclax. Navitoclax is now in phase 2 clinical trial, and this is active on the both BCL2 variant as well as BCL-XL. So to make it more selective, further the fragment-based drug design is used, which lead to the venetoclax, which is also very important anti-cancer drug and is approved by the FDA. And this is very much active in comparison to the navitoclax. Therefore, this is said to be BCL2 selective inhibitor, having the activity less than 10 picomolar. So this is one of the interesting example where fragment-based drug design success is being dramatically lead to the discovery of the drug molecule. So earlier I discussed about the different approaches for the fragment-based drug design. And generally in this case, optimization or characterizations are being done initially on the basis of the experiments or experimental design, like say using NMR, ITC, XRD, or so many technique can be used over there. But those are somehow cumbersome and require the expertise. Along with those are time consuming also. So nowadays we are turning toward the more easiest approach, that is a computer-based fragment drug design. So in this case, initially structure-based drug design softwares are being used, and those are like say Grid, MCSS, Ludi, Sprout, Superstar, Music, Arico are very useful software which are based on the structure-based drug design, or we can call as a de-novo drug design. So de-novo drug design, that is also based on the fragments. So initial fragments are need to be identified in all these approaches, and one or two different algorithms are being used in these different software. So like say in Grid, ligand binding pocket mapping is being done with a molecule probe. In Ludi, commonly the hydrogen bonding as well as hydrophobic interactions of the fragment are being used in the concentration. Sprout, where primary and secondary characteristic of the receptor are being used, where we have to identify first the binding pocket and the possible interactions of there. So there are a lot of computerized structure-based drug designs being used earlier and nowadays also. Those are based on the fragment-based drug design approach. But purely there are some programs which are used nowadays based on the fragment-based drug design is ACFIS server. This is freely available and one can upload the query online and get the results in 1 or 2 days. Then the another is Auto TNT, Charming Web User, eLead 3D, eMolFrag, Lick Builder 2, FluAx, MolBlocks, as sample and such. So there are different programs are available for the fragment-based drug design, and those are helpful in the later on finding out the lead molecule using the easiest approach by the computer system. So here, to synthesize or do the experimental work, but later on, of course, once we identify the hit and hit to lead, then thereafter the experimental studies can be carried out. As I was discussing earlier, the most important part in the fragment-based drug design is the fragment. And I told you how we can define the fragment. So generally rule of 3 is being used. So like molecular weight, we have to take care. Then hydrogen bond donor, acceptor, along with the polar surface area. Okay, log P. So all these need to be considered while selecting the fragments. So these different fragment library can help you to select the different fragments. So they have a databases over there, and based on the need, we can select a particular type of fragment. So this is a table shows you the different fragment library and the different resources over there and their website or the link. So you can use the 3D Fragment Library Consortium, which has a great shape and diversity analyticon. This is particularly for the natural product. The natural product fragments are available there. SNX, Cambridge, which is having over 6,000 compounds following the rule of 3. Chem division, Inamine. So all are having the fragment which are following the rule of 3. Some commercial, most of them are commercial, so like the Jinka can also be used over there as a commercial library where millions of compound and fragments are there. So somewhere you will find the molecules also, not a fragment. So that can also be used as for the primary screening process. So I'll basically start with a few software which can help you in designing the lead molecule or lead compound using the different fragments or generating the fragment itself. So one interesting example of the software is the LUDI, which is the DNO drug design based software. And this works on the basis of the fitting of the fragment at the different region. You can see here, if this is the particular protein and it has a different site or region in the binding pocket, these binding pocket are being identified based on the possible hydrogen bond donor, acceptor, or hydrophobic areas. Okay, so here you can see the carbonyl, which is there in a binding site, and carbonyl is the hydrogen bond acceptor. Here you have a NH means amino, which is the hydrogen bond donor. And the area where like say phenyl or something like say alkyl substituents present in the binding site are moreover reflecting the hydrophobic area. So identifying is the first part. So identifying the interaction, possible interactions at the binding site, then putting the fragments, fitting the fragment at the particular binding region of the binding site. So if this is the hydrogen bond acceptor, then we have to put the molecule or fragment which is having the hydrogen bond donor. Same for the hydrophobic, then you have to put the similar hydrophobic fragment. And vice versa for the hydrogen bond donor, you will be fitting the fragment which is containing the hydrogen bond donor. Okay, so this is initially identified by fitting the fragments using the molecular docking studies. So one and another things are being connected together in the fragment-based drug design approach. And later on, those are being bridged at the final stage with the appropriate linker. So this is called as a linking approach. Now, this is actually being done on the basis of the identification of the binding site initially. So as I told you, if any alkyl substituent or aromatic substituents are there in the binding site, then the hydrophobic area need to be constructed. That may be near around 4 angstrom. So that sphere need to be constructed. Then the sphere is need to be identified for the possible interaction. So at a lower side, you don't have the probability of the interaction, as a hydrophobic interaction. Therefore, you will be trimming the area, and only this is the exposed position or exposed area where the interaction is possible. Similarly, as this is the non-directional interactions, like say van der Waals or hydrophobic interactions, so therefore there is no need to specify the directions. But in the case of the hydrogen bond donor as well as hydrogen bond acceptor as well as hydrogen bond donor, as these are the directional bonds forming the hydrogen bond, and therefore we need to define the directions. So in case of the binding site is having the carbonyl, then there are two possibilities from left and right side. And if the binding site having the hydroxyl functional group as the hydrogen bond donor, then it has the one probability. So bridging is being done based on the some logical approaches, or linking is being done. Initially, if you have a two fragment at one region and another region, the close proximal hydrogens are being identified first, and those are being replaced with the some linker which is appropriate in the distance. So it could be carbon-carbon single bond, it could be carbon-carbon double bond, and that is being defined based on the distance between these two connecting atoms. So linker has a lot of probability. It can be simple carbon chain containing the single bond, double bond, triple bond, or the heteroatom containing linkers, or the different connecting function groups linkers like say sulfonamide, amide, amine, ether, esters. So all possibilities are there, and that is being optimized later on by finding out the affinity score of the lead molecule. Then another program is this Sprout. This program is also based on the same fragment-based drug design. So this is based on the link and groove. But here initially fragments are not being fitted. Initially all possible interactions are being identified, and a sphere nearly same size of the atom, particular atom, is being placed over there. So say example here, if we have the hydrogen bond donor, then we will be putting the hydrogen bond acceptor, and that hydrogen bond acceptor could be the oxygen. So we will be putting the sphere which is mimicking the oxygen over there. And then that will be grown in other area. So same if here we find out like say aromatic, like hydrophobic interaction or van der Waals interactions are there, then we will be putting the aromatic type of the connectivity, not exactly the benzene ring. Okay, it's initially a sphere, atom spheres are being added. So all later on are being transformed into the skeleton. The skeleton having the possible connections of the atoms, and those atoms are being specified. Somewhere these are being specified, like say here for the hydrogen bond acceptor atoms. Okay, here for like say hydrophobic interaction, and the another side where you have the again hydrogen bond acceptor. So at last these are being converted into the structural molecule by joining the dots. And these dots are being joined based on the distance between one and another possible atoms. Like say this is one example where we actually come up with some molecule which has the single bond connection, again. Then later on the like say sp hybridized carbon possibly connected with a sp2, so it means there must be double bond, and then sp3 atom. Okay, so this lead to the generation of this enol, which is having the double bond over here and adjacent as a hydroxy. But this enol is, we know that it remain in a tautomeric form, and rather than enol, it exists in the form of ketone form. And that lead to the drastic difference in the binding mode of the designed molecule or the fragment. So here hydroxy is acting as the hydrogen bond donor, and once it converted into the keto form, it becomes the acceptor. So that is a drastic difference can be observed. So this Sprout software allows you to modify the other possibility, like say, rather than this enol, we can transform it into the carboxylic acid. So that will remain hydroxy as such. There will be no tautomerase between these two. Also, it helps in the modification where we can construct the molecule easily. Okay, like say here, the connection of the two cyclic chain would, with a 2 carbon linker, would be difficult. Instead of that, we can put the heteroatom or particularly oxygen which can later on easily synthesize. So this is an example how we can easily synthesize this designed molecule by just using the salt of sodium salt of this 6-member cyclic structure and using the SN2 reaction, substitution reaction can give you the design model. So this software also look at the possibility of lead molecule which can synthetically feasible. Because ultimate role for this is to synthesize and evaluate the molecule for the pharmacological screening. I'll be coming out to explain you the another important fragment-based drug design software, and that is freely available. One can go onto the server and can use it directly to design the lead molecule out of a given ligand. So this flow chart shows you how this ACFIS, that is the auto core fragment in silico screening, usually works. You have to give the input of the protein ligand complex and the cofactor parameter file. So protein ligand complex will be having the ligand itself. So at initial stage, the properties of these are being calculated based on the parameter provided, which is minimized in energy, and the ligand which is available in the binding site is deconstructed. So that is being defragmented, you can say, or deconstructed means using the retrosynthetic analysis approach. This is being chopped down into the smaller fragments, all possible smaller fragments. Then those fragments are being reassembled to find out the other novel molecule. So those may be having the similarities with the initial fragment or may have some diversity. So all these are being further screened virtually, and binding energy is being calculated. And this binding energy is being compared with the initial ligand to know whether we got the new hit or the lead molecule or not. So this is very simple. In the next slide, in this slide, you can see how this works. We have to give the input for the protein ligand complex. So it means we must be having the ligand as well as protein for this particular fragment-based drug design approach. So once you submit your job is being confirmed, and later on, stage by stage, the program is going to evaluate the molecules and give the result. So these results are going to show you the binding energy basically, and along with the binding modes as well as the different molecular properties. These different molecular properties help you to identify the lead molecule, whether this is particularly showing the higher affinity, is feasible in other aspects also or not, like say low p-values or the other issues, or a better fit probability also being determined over there. Not only this gives you the idea about the synthetic feasibility. So whatever fragment are being connected by this program lead to give you the lead molecule, but that lead molecule, whether that is feasible to synthesize or not. So it will be using the retrosynthetic analysis, and based on that, to give the possibility of the synthetic event. Nowadays, we have the evolutionary approach, which comes in the fragment-based drug design. This evolutionary approach is somewhere where we were discussing, like say, defragmentation or deconstruction of the fragment itself, molecule itself or ligand itself, and rejoining or reconnecting. But in this evolutionary approach, here is the key point which actually differ from the other approaches. So I'll explain you the algorithm, how this fragment-based molecular evolutionary approach works. Same as I discussed in the previous slide, we have to give the reference molecule, but here protein is not necessary. Okay. So initially we have to give the reference molecule. Then this is being transformed into the seeds, and these seeds are the, like, so you can see in this figure, this initial reference molecule is deconstructed or fragmented into the different fragments. Okay, and these fragments are having the different like the ring structure, linker, or side chain. So out of these, seeds are being generated. Seeds means these are the initial point. So these are considered as the seeds. Then these are being again converted into the initial structure. Means some fragment are being joined together to find out another structure. And that is done later on the basis of the mutation or crossover. Mutation means change in the particular position with the other ring or the other side chain. Crossover were completely changing the one side with another side. Then these are being evaluated, all molecules will be evaluated by the fitness score, Tanimoto coefficient, which shows you the biologically similar molecule. Then those are being selected and compared. If we get the right kind of designed molecule with a better Tanimoto coefficient, then we stop. Otherwise, we will repeat this process. So this will lead to the diversity in a molecule. Okay, so you will get the complete different structure from the initial reference molecule. Now, in this approach, we have a general thumb rule, which are allowed connection and prohibited connection. So allowed connection, where ring-ring connection are allowed with a side chain, of course. A ring linker connection with a ring is allowed, okay, because this is a linker, so that must be in between of the two rings. A ring side chain connection is also allowed. So you have a ring, side chain ring, linker, ring, okay. So all these three possible connections are being allowed: ring-ring, ring linker, or ring side chain. But these are the other possibility. Okay, you have another three possibilities, but those are not allowed because of the lack of the stability or synthetically unfeasible or difficult in synthesis. So linker-linker connection. So linker-linker connection, this kind of linker-linker connection is not useful and it's difficult to synthesize as well as not appropriate. Sidechain is also not useful. Linker sidechain. Okay, so linker sidechain is also not useful. Therefore, these three connection approaches are being prohibited. So this is one of the example you can see. So you have a base fragment. So base fragment is being replaced with another fragment. So this is one of the fragment is identified and is being removed. And other terminal possibilities. So this is one possibility, another possibilities. So that lead to the newer design scaffold. Also, you can replace the linker between the two rings. So this is this ether linker. So you can replace with a amide or similar kind of linker. So it means that is totally mutation where you will be changing a particular fragment with another one. Now, another approach is the crossover. In crossover, you suppose you have two molecule and two fragment, okay, and those two fragment where you have a specific particular fragment is being replaced with another. Crossover means you will be replacing one fragment of the one molecule with another. So that is another approach which lead to the progeny or child molecule. So from the parent, after getting it crossover, we will be getting the newer molecule is somehow differ in the connectivity as well as in the structure. So these approaches are very much evolutionary and lead to the diverse set of the compound. So this is one of the example for the ligand design using the fragment drug design approach, where one fragment is being selected, one molecule is being selected as the reference molecule from the Cambridge library, okay, and that is being fragmented into the seed fragments. Then molecular evolutionary approach is being used. So different runs are being made. One run to, or maybe here almost different possible replacement are being made on the molecule, and all are being compared with the Tenimoto coefficient. Okay, so Tenimoto coefficient will tell you what exact similarities are there from the reference molecule or not. And this tells you the better fit at the binding site. So generally, if Tenimoto coefficient is more than or near around 0.8, will be considered as the best fit, and lesser than that would be considered as the worst fit. So in this case, you can see here, this molecule is not fitting and having the Tenimoto coefficient is 0.57. So other are being having the better Tenimoto coefficient, and that will give the idea for the development of the active molecules. This is one of the molecule is being developed having the Kd dissociation constant in around 14 nanomolar. So using these approach, computer-based drug design approach, where different fragments are being either linked or grown or used via the help of the evolutionary approach are under clinical trials. These are the few examples shown in the table. Fragment-based drug design approach, where there are a lot of molecules, as I told you, almost 30 molecules are under a clinical trial. 3 drugs are being already approved by the FDA. Okay, so these are the 3 drugs: Vidafitinib, Vemurafenib, Ventoplex. All 3 are being approved. So all are being designed as well as discovered with the help of the growing and linking approach. So different other examples are also there, where different pharmaceutical companies are working on the basis of the fragment-based drug design approach, and these are the different molecules which are currently under the clinical trials. So there are a lot of molecules are under the clinical trial, phase 2, phase 3 clinical trial. So it means that shows the potential role of the fragment-based drug design in the development of the hit to lead molecule and thereafter lead to drug molecule. So at the end, I'll be going to summarize the today's talk. That fragment-based drug design, as we discussed, is the novel approach for the designing of the lead molecule from the hit molecule. It also suggests the different binding modes, new binding sites. We can use the different approaches, which is based on the experimental work as well as the computational work. It uses the high amount of the structural data. It requires the small time or lesser time in drug discovery process. Also, it has a high rate of the hit to lead success ratio. And one important thing that it lead to the novelty as well as diversity in the drug design of the fragment to the lead molecule. So that give you the exact potential how this fragment-based drug design is very useful, particularly in current scenario where we are actually searching the drug molecule for the treatment of the COVID-19. So fragment-based drug design can also help in that particular area where we can accelerate the drug discovery process by getting approved the drug in the lesser time period. So with this all, I'll be thanking you for your kind attention, and I'll be going to acknowledge the efforts of the drug discovery hackathon team and our director, Dr. S.J.S. Flora, who inspired me to involve and to get in such kind of noble cause where nation is trying to strive for the finding out the drug molecule for the treatment of the COVID-19. So I hope we will be going to succeed in this area soon. With this, I'll be thanking you all. So any question or query?
Yes, thank you so much. It was really a very detailed presentation over the topic. So as we have a little bit time constraint, so I will quickly take few questions. So the first question arises as how fragment-based drug design is different from de novo drug design.
So this is a very interesting question, and I focused on the same aspects in while I was presenting. So fragment-based drug design approach is the key approach which is used in the de novo drug design. De novo drug design, where I told that this is totally based on the uh, protein structure. So de novo drug design generally used when we have only information regarding the protein structure, not about the reference molecule. But in this case also, the fragment-based drug design help you to find out the lead molecule, because these fragments are being used there in the de novo drug design, are being uh, utilized from the fragment libraries. So those fragment libraries must be following, as I told you, rule of three. So therefore, I must say those are differ from each other, but in broader way, fragment-based drug design is approach is used in the de novo drug design. So it's a part of the one of the part of the de novo drug design.
Thank you, sir. Another question which comes up is how to determine which conformation of fragments should be selected to form a candidate.
Yeah. So therefore, we discussed earlier also. Generally, fragment-based drug design approach, where fragments are being selected on the basis of their planarity. Okay, so they must not be having the too many possible conformations, possibly only one conformation. That's how we used to consider those fragment based, which are having the lack of the conformational changes. So those fragments which are having the low conformational changes or no conformation changes, those are preferred in the fragment-based drug design.
Fine, sir. So I will take a last question. It is related to the tools. When we have a protein as a start, what are the best tools to identify candidate ligands?
So tool, when we have a protein, then I told you once in one of the slide, I discussed, yeah, this one. So in this case, generally I told you they are being categorized into the two broad category. One is ligand based, another is structure based. So the question is, when we have information or structure of the protein, then we can go through the molecular docking study, one of the approach, and de novo drug design. That is another approach.
Okay, fine. Thank you so much, sir. So as we come and end to this session, on behalf of the chairman of this DDH training program, Dr. G.N. Sastry sir, and the entire team of the DDH, I'd like to thank you for giving such a wonderful presentation. And also I'd like to thank all the attendees who were there in the YouTube and MS team for putting their questions. So thank you so much, sir.
Thank you so much. Thank you all.
Thank you. So we'll wind up this session here, and our next session for vertical tree will start at 6 p.m. Thank you.